Stroke is a leading cause of neurological disability and death worldwide, with neuronal damage being the primary determinant of clinical outcomes. One of the central mechanisms underlying neuronal injury after stroke is glutamate excitotoxicity, a process wherein excessive extracellular glutamate leads to overactivation of glutamate receptors, particularly NMDA and AMPA receptors, on neurons. This overactivation results in massive influxes of calcium ions, triggering a cascade of deleterious events, including mitochondrial dysfunction, oxidative stress, activation of proteases, and ultimately cell death. Under normal physiological conditions, glutamate acts as the main excitatory neurotransmitter in the central nervous system and is tightly regulated by uptake mechanisms involving astrocytes and neuronal transporters. However, during ischemic stroke, the deprivation of oxygen and glucose impairs ATP-dependent processes, leading to the failure of glutamate reuptake and accumulation of glutamate in the synaptic cleft. The resulting excessive stimulation of postsynaptic receptors not only disrupts ionic homeostasis but also activates cell death pathways, such as apoptosis and necrosis. Experimental studies have demonstrated that interventions aimed at reducing glutamate release, enhancing its uptake, or blocking its receptors can mitigate neuronal injury in animal models of stroke. Despite these findings, clinical translation has proven challenging, in part due to the complexity of glutamate signaling and the risk of interfering with essential physiological functions. In summary, dysregulation of glutamate signaling plays a pivotal role in mediating neuronal damage following stroke through the mechanism of excitotoxicity. Continued research into targeted modulation of glutamate pathways holds promise for the development of neuroprotective therapies that could improve outcomes for stroke patients.